me s- Let me start by welcoming our speaker, Harris Lewin. Uh, uh, um, Thank you very much for joining this NF Core special interest group dedicated to animal genomics, and thank you all for joining. This session is being recorded, and before giving the floor to Harris, let me very briefly introduce him. For many of you in this channel, Harris does not need much of an introduction because he has had a long career of geneticist of farmed animals. And Harris' alma mater is Cornell University, where he earned his bachelor in animal science in 1979 and then did animal breeding and genetics in 1981. He was awarded a PhD in immunology from Davis, University of California in '84. Harris is especially well known for his work on bulls, and I was reading this in Wikipedia, Harris, which I didn't know. So apparently, you discovered solved very interesting mysteries about breeding and figuring out about some inbreeding that had very, very serious consequences in the bull population. And of course, you are very well known for your work on cow. Uh, um, you have also had a very long interest in a fascinating topic, which is the evolution of chromosomes and why do we have so many chromosomes? How do they increase in number, decrease in number? All of these things, which I understand or hope you may give us a little bit of insight. But you will also be talking about this extraordinary journey on which you have recently embarked, which is the Earth BioGenome Project, of which you are the chairman. And this project is by far the most ambitious project ever taken by biology, with 1.8 million eukaryotic genomes planned to be sequenced in the near future. You know, there's no a single example in science in general and in biology in particular where more data did not lead to a change of paradigm, and you are leading this effort, which is something amazing. So we are extremely honored to have you here with us today, and I will now stop talking and giving you the floor. Harris will talk for about 45 minutes and will then be available for your questions. So without any further ado, let's welcome our guest. Thank you, Harris.
Thank you very much, Cedric. Give me a minute here.
I
Get
forgot to say that
my
Harris is also a guitarist in the Wealth of Nations and quite a famous musician. So I encourage all of you to Google this online.
Well, thanks for that plug. That was the first time, and yeah, it's really great to be here. It's early in the morning here in California, and I'm extremely pleased to be with this group. I understand it is an international group, so there are folks probably from around the world. The coffee hasn't quite kicked in yet, so if you'll give me a few minutes, I'm going to warm up. I appreciate your introduction very much, Cedric, because I am going to go back and do a bit of a retrospective today since this is an animal genetics interest group and focus group. I'm going to first give an overview of the Earth BioGenome Project, and then I'm going to tell a story uh, about how genome sequence information and resequencing and comparative genomics led us to the discovery of this very important mutation, which has caused more than 500,000 abortions worldwide in dairy cattle. So I'm going to talk about that today. So I haven't done that in more than 15 years, that talk, so I had to go back and dig some slides out. But so that's why my subtitle is Tall Tales and Semen Trails for today. I hope you'll all enjoy it. Okay, so hopefully I can advance. It doesn't look great. Okay, so as Cedric mentioned, one of the main activities that I've been involved in the last 10 years is the Earth BioGenome Project. I co-founded this project along with Gene Robinson, the University of Illinois, an entomologist, and John Kress at plant ecologist at the Smithsonian Institution, and Huan Mingyang, who is the president of BGI in China. And the Earth BioGenome Project is now a very large international network of networks that has this common goal, very ambitious goal, as Cedric pointed out, of sequencing and annotating the genomes of all 1.8 million named eukaryotic species in 10 years. And for those of you who haven't seen it, the manifesto for this project was published in 2018 after 3 years of thinking and planning and trying to understand how we could conduct such an ambitious project across the globe. And basically, for you, bioinformaticians and computer scientists out there, the main goal of the project is to create a digital repository of eukaryotic life by sequencing all the named eukaryotic species. And this collection of sequences would then represent sort of the collective wisdom of 3.5 billion years of the evolution of eukaryotic life on Earth. It would also be foundational for scientific efforts understanding the origins and evolution of eukaryotic life on Earth, as well as providing a foundation for phylogenetics and functional genomics and synthetic biology and several other disciplines that are important for the advancement of the global bioeconomy. And we recognize now that biodiversity loss, so you say, what is the rationale really behind this project? And we recognize that the loss of biodiversity poses as great a risk to humanity as climate change. In fact, the two are intimately linked. We understand that now that we can lose up to 1 million species by the end of the century if nothing is done to reverse the effects of climate change and reduce pollution and overexploitation and the many other anthropogenic factors that are influencing this very rapid increase in extinction rate that we're seeing across the planet. And the reason why this loss is concerning, I mean, losing 1 million species, of course, could lead us to a tipping point, and that tipping point could very well affect the ecosystems on which we all depend on this planet for our own lives and our sustenance. And so biodiversity provides the basis for all of the ecosystem services that support life on Earth, including agriculture, which we're going to discuss today, clean water, medicines from nature, and regulation of Earth's biogeochemical cycles. And the these ecosystem services provide the foundation for the global bioeconomy. And the global bioeconomy today is in the trillions of dollars. When this slide was made, it was in the hundreds of millions. But now, globally, we're probably at about $2 trillion a year, and that is rapidly increasing. And the bioeconomy will be a very substantial fraction of the total GDP on the planet in the next 20 or 30 years or so. And so these are the main driving factors that are currently leading us and providing funding for this major project that we have embarked on. Now, what does the Earth BioGenome Project look like? Well, today we have 58 affiliated projects around the world that are based in 18 countries. Those 58 projects in 18 countries actually expand if you look at all the participants. So nearly half the countries of the world, 88 countries, are involved in one way or another, either from sample collection to sequencing, analysis, annotation, and so on. And the project, as we can measure it today, involves more than 2,000 participants. The project is committed to open data access, compliance with the Convention on Biological Diversity and the Nagoya Protocols on access and benefit sharing, and also committed to the principles of diversity, equity, and inclusion within the EBP community of scientists and with indigenous peoples and local communities. Now, as you can see on the right here, the project is organized as a sort of a hub and spokes model. If you look around the outside in blue, those are the 58 individual affiliated projects. Many of those are taxon specific projects. Some of those are projects that are geographically based. We are now in the process of organizing these 58 projects under a system of regional nodes. And the regional nodes will correspond to broad biogeographical areas around the planet. Roughly today, the first set will probably correspond to the continents, except for Antarctica. And we have just launched this process this week, in fact, for applications for the regional nodes, which will then coalesce all these individual projects on a regional basis. Now, at the center of all this is the Earth BioGenome Project Secretariat, which is currently located at the institution where I currently have an appointment. I've retired from UC Davis and have an appointment at Arizona State University, and Arizona State University is hosting the Secretariat. And the governing body for the Earth BioGenome Project is the Earth BioGenome Project Executive Council, which is a 8-member council, 9 including me, that is elected from the EBP membership. The Executive Council is responsible for the development of standards for the project. That is the standards for the collection of samples, processing of those samples, standards for sequencing and assembly quality, standards for annotation, standards for analysis, and standards for IT. There are also committees in the EBP that are developing policies related to the membership, related to intellectual property and data access and sharing, and all of this is codified under a governance document, which we adopted formally in 2023 after spending about 8 years or 6 years under a memorandum of understanding. So I just tell you this just so you can understand the scale and scope of current scope of the Earth BioGenome Project as it stands today. And as I mentioned, the projects are scattered. These 58 projects are scattered throughout the world. You can see there's already a very continental distribution of these projects in Asia, in Australia, in Africa. In fact, the Africa BioGenome Project, which is headquartered in Kenya, actually has 22 countries that are involved in Africa BP. There are projects in South America, in Colombia, in Brazil, in Chile, and several projects in North America. And all of this is being coordinated under the umbrella of the Earth BioGenome Project and through the Earth BioGenome Project Secretariat. Now, in the 2018 paper, we discussed how this project would be executed. And originally, we talked about three independent phases. In the first phase, we would sequence a representative species from all 10,000, approximately 10,000 eukaryotic families. Phase two, and that would be in the first 3 years of the project. Phase two would be a representative of each of the genera, and phase three then would be the remaining species. We had a meeting in 2023 and we revised those objectives to be more in line with what the individual sequencing labs are and projects are actually doing, and to be more in line with the goals and expectations of the funding agencies that are sequencing the project. So these are the revised objectives. This paper, which will describe the results of this workshop that was held in Lausanne in 2023, is just about to be accepted for publication and it will be published in Frontiers in Science. And I'll just say that the revised goals are then it's still to sequence about 10,000 species in the first phase, but only 50% of those would be representative of each eukaryotic family, and 5,000 would be other species, could be other species within those families, and that is just the way the project is proceeding. And I'm going to show you progress toward the phase one goals in just a few moments. And these phases are now not discrete, they kind of overlap. And so in phase two, we'll complete the sequencing of a representative of each of the eukaryotic families. But in addition, we're going to target 50% of the eukaryotic genera. Okay, so that's about 50% of 134,000, then topping that up to 150,000 species in 4 years. And the real guts of the workshop that we had in Lausanne and the details in this paper are the strategy of how we're going to go about doing 150,000 genomes in 4 years. This is a major challenge from where we are today and require scaling from where we are today more than tenfold. And so this is going to be a huge challenge to get this done in 4 years, but we think is possible. And then in the out years, in the remaining years of the project, to sequence the remaining species. And of course, we're nowhere near yet having the being able to produce genomes at that scale. And we're certainly a long way to achieving funding for that, but we are certainly well on our way in phase one and phase two. And so let's look at this global status of eukaryotic genome sequencing around the world. And this is EBP and the rest of the world. So it's not just EBP. If you go to INSDC databases, you will find that now there are upwards of 19,000, nearly 20,000 distinct eukaryotic taxa with whole genome sequence available. And that's as of just this week. I know the bottom says February 3rd, but that should be February 17th, as it says at the top. But as you can see, there's really a long way to go in this project, as 19,500 species only represents about 1% of the described eukaryotic species. So everything we know, everything we think we know, is only based on a very small fraction of eukaryotic species that have been sequenced. So there's a long way to go, and there is a lot to learn as we go. If you look at the phylogenetic distribution, this is the, just sort of these rainbow chart is a summary of how the species that have been sequenced are distributed in the different phylogenetic levels. So even if you look at the phylum level today, there are 67 eukaryotic phyla, and we don't even have a representative species sequence for each of the eukaryotic phyla. We're still missing about 12 of those, but progress is being made. And if you go down that list, the area that we've targeted for phase one is at the family level, and already there is at least a genome sequence of some quality for nearly 30%, 25% of the eukaryotic families. However, uh, if you look at the quality of those sequences, quality of those assemblies, only 1 in 5 meet the standards that have been set for the Earth BioGenome Project. And so that means that 80% of what's already been done, like 16,000 or so species, are probably going to have to be redone in order to do the kinds of things that all of you would like to do in the kinds of work that you're doing with ENCODE. I mean, we cannot work with highly fragmented assemblies that are missing big regions of the genome or have very low quality in terms of the base calling accuracy. Okay, and so what is the Earth BioGenomes Project's contribution been to these 19,500 species? Well, so far, the EBP, under the EBP umbrella, which is given there, has the EBP affiliated projects have produced, now we have passed 3,000 unique species, which is really a major accomplishment for the project. So we are already more than a third of the way toward the phase one goal that we have set, and we are on track to complete phase one based on the funding that's in hand and the samples that are in hand by the end of 2026. And so I need to put a caveat on this, and that is that this is an underestimate of what EBP is actually produced, because not all of the EBP affiliated projects have been integrated into our tracking system, which is called GOAT, or Genomes on a Tree, which was created by the Wellcome Sanger Institute. And so I think it's probably off by about 20%. For example, we don't have JGI in there. We don't have the BGI genomes integrated into this counting system yet and tracking system. So these are underestimates. We're probably, well, we know already that we're well over 4,000 in unique species from the existing EBP affiliated projects around the world. So 3,319 unique species representing 2,459 genera and 1,115 families, more than 10% of the families, taxonomic families, have a representative sequence. If we look at how that breaks out by phylum, you can see that most of the species, that darker purple bar on the left, are arthropods. So nearly half the species sequence to date are arthropods by EBP. About 1,000 of those are chordates. 236 are plants, and so on. Most of these other phyla have very low representation. I point this out because, the area of the tree of life, the parts of the tree of life that are most understudied still represent the greatest amount of eukaryotic diversity, and that's the protists and the single cell eukaryotes and the small things that we find it hard to collect, measure, see, and classify. And so this is a huge area of scientific opportunity, and for all of you who are working in this space, these are really interesting organisms, and there's lots to do and some very exciting new techniques that are allowing very high quality genome assemblies from very small numbers of cells, under 100 cells, and even now methods that are producing at least decent drafts from a single cell. And this will have a very, very big impact on our understanding of the origins and evolution of eukaryotic life. Now, we're in a great position because we have been doubling. This is the year over year, so I gave you the actual numbers. You can see that we've really doubled in the last 2 years in terms of species, and that most of those species of that lighter blue color there, you can see those are the chromosome scale assemblies, where that darker blue are the scaffold assemblies or contig assemblies. So the EBP is really making great effort to produce assemblies that are meeting our EBP minimum quality standards. And those are the ones that are the fastest growing and doubling quite rapidly. And this must increase in order for us to achieve our goals. And the statistics that I guess I'm most pleased with and most proud of for those that are producing these genomes is this contribution to the overall number of reference quality genomes that have been produced around the world. So these are the minimum metrics here. The metrics are much greater, but the minimum metrics for these assemblies of contig N50 of 1 million bases and scaffold N50 of 10 megabases. And if you look at that, as I mentioned, there are about 20% of that 19,500 that meet the EBP, minimum EBP standard. So there's about 4,000 of those. And of those 4,000, 2,236 have been produced by EBP affiliated projects. So more than 50% now of reference quality genomes have been produced by EBP affiliates. And if we go over to the far right, you can see, I think, what is one of the most impressive statistics that we have today for the project is that nearly 70% of the reference quality genomes at the family level have been produced by EBP affiliated projects. This is really an extraordinary accomplishment. And again, these are underestimates because not everything is in the GOAT system. This is looking by affiliated projects, and it's a little bit, misleading. We have a few places in the world that are really leading this effort, and I have to give a shout out to Mark Blackster at the Wellcome Sanger Institute for his leadership of the Darwin Tree of Life project, and Eric Jarvis at Rockefeller University in New York, who's been leading the vertebrate genomes project. And these two projects have produced the majority of sequences of EBP quality assemblies that we have to date. As you can see, there's differences in terms of what people are focusing on. Some are focusing on plants, some are focusing on, as I mentioned before, Darwin Tree of Life has done lots of insects, particularly focused on lepidopterans. Some are working on fungi. But it's a little bit, I guess I don't want to undervalue these smaller projects like the California Conservation Genomics Project, which has produced 150 reference quality assemblies. That's no small contribution. In fact, that has really been one of the most important flagship projects we have in the conservation space. But Darwin and VGP have really done an amazing job and are leading the way. And we have, again, between Darwin, VGP, and these other projects, funding is in place and samples are in place to get to the 10,000 genomes in 2 years. So this is an article which appeared, a little news story that appeared in Science after the biodiversity genomics meeting last fall. And once thought a fantasy, the effort to sequence the DNA of millions of species gained momentum. We are definitely on our way to this. There is no turning back. I am very confident that we're going to complete phase one now by 2026. And I think we have all the pieces of the strategy that are necessary to do 150,000 genomes over the succeeding 4 years. So we're well on our way, and it's very exciting to be part of this, and I'm very privileged to be leading this effort, especially at this later stage of my career. But since I am sort of an old timer, and you all are interested in animal genetics, and so I'm going to wind back the clock to what got me interested in all of this. I started back in my research career 45 years ago, as Cedric mentioned, at Cornell in the late 70s and early 80s. And since the 80s, I guess we can distill down the questions that I was interested in, at least for the first 25 years of my career, were how do you improve this? This animal that has been highly selected over multiple generations for its ability to transmit genetics, high quality genetics for milk production and milk production traits. And that's was sort of the question that got me into all this from the beginning. And I'm going to tell you a brief story. I only have a few minutes, so I'm going to have to skip parts of this story to get through this. But just to make it, to make it clear, this strategy, which we talked about after the cattle genome sequence was published, and as all of the other livestock species were being developed. This is pretty well encapsulates what we were about and what you folks are currently working on, and that is at the bottom of the pyramid to really understand and to get very high quality comparative and evolutionary analysis of livestock genomes, which would then allow us to do a complete dissection of the genetic elements, genomic analysis for complex traits. We were able to do systems biology, and all of these combined techniques would feed into the development of new technologies for improving animal production and animal health, such as genomic selection, such as transgenesis, now with CRISPR, and biopharmaceuticals, and all of these things are now driving. In fact, when we started, it was only statistical animal breeding, and now molecular animal breeding has sort of taken over in many ways or has enhanced the methods that were used for the first 50 years or so of animal genetic improvement through quantitative genetics. And so, again, my own story starts back in the 80s. And I'll just say that we started with a single locus, and that locus was the major histocompatibility complex. We had no genetic markers. We were typing using classical immunogenetic techniques. This was serology based in the beginning in the 80s, and then moved to genetic markers and sequencing. And the really first, I think, important discovery in our group was the discovery that genetic resistance mapped to one of the class 2 genes of the major histocompatibility complex, and we could identify 2 amino acids in the DR beta 3 chain, one at position 70 and one at position 71, which were right in the antigen binding groove of the MHC class 2 molecule, which were responsible for resistance to the subclinical progression of infection by bovine leukemia virus. So the animals could be infected, but once they were infected, if you had the resistance allele, resistance was dominant, and you would not progress to the next subclinical stage of infection, which was known as persistent lymphocytosis. Well, when I was an early assistant professor, one of the chairman of the Department of pathology in the vet school at UC Davis at the University of Illinois told me, he said, a mouse is not just a chromosome 17 with hair. And so I took that lesson and I said, well, a cow is not just a chromosome 23 with an udder. Uh, we were very limited on what we can do, and that got us started to thinking about expanding into the rest of the genome. How could we do this? How could we map traits? How could we find genes that control complex traits in an outbred population? And that we immediately began to understand was going to be dependent on the development of high quality genetic maps. Over the years, my lab was involved in a number of these maps. Several people on the call I see have as much gray hair as I do and will remember this era of mapping, which came before sequencing. We started with linkage maps, produced the first male linkage map in '96, then switched in collaboration with Jim Womack to radiation hybrid mapping. The first generation comparative maps were 760 markers in 2000, and then 1,400 markers based on a new strategy that we developed called COMPASS, which allowed us to do comparative mapping by annotation of cDNAs and sequence similarity. And then finally, using back-end sequences that we can map the ends of those back-end sequences and select markers that were evenly spaced across the genome. In the 2005 map, which Dennis and Annalee produced, Annalee Everts-Vanderwin produced, contained about 3,000 markers on this radiation hybrid panel, or markers about 1 million bases distant from one another. Of course, this map was extremely important in the early days of sequencing. We were not producing the quality of genomes that we are producing today. We were producing highly fragmented assemblies. The first cattle genome assembly was published in 2009. But these maps, the map was absolutely critical for the assembly and scaffolding of the cattle genome. So that 3,000 marker map was the map that was used for the assembly of the first draft of the cow genome. And then, of course, that opened entirely new doors into comparative genomics for us. And an example from that paper is just showing the long arm of human chromosome 1, that we could now do synteny analysis at great detail and identify evolutionary breakpoints in chromosomes all over the entire evolutionary span of the mammalian lineage. And I'm not going to talk about that today because I only have a few minutes left. I'm going to talk a little bit about mapping traits because we were involved in doing that way back even in 1990, one of the first studies done not with a single genetic marker, but with biochemical polymorphisms and blood groups, believe it or not, in 1990. But I want to point out this study because in only 9 years, we were able to collect the largest, make the largest collection of semen samples in the world at that time for QTL mapping. It was called the Dairy Bull DNA Repository, and it was used for mapping QTL by genome scanning in the North American Holstein Friesian population. I tell you that because this collection of semen, this is part of my Semen Trails title, this collection of semen proved to be very, very valuable in identification of important traits. So 2 of the bulls in this collection were among the most elite sires in the history of the Holstein Friesian breed. And when sequencing became less expensive, so to produce the first cattle genome, that draft sequence, which nobody uses anymore, cost $50 million. But by only a few years later, 5 years or so, 6 years or so later, there were new technologies, and we used 454 technology to resequence 2 of the most famous bulls in the Holstein Friesian lineage. And the idea was to look for long-term signatures of selection for milk production traits through a process of sequencing and genotyping, and I'll describe that in a moment. So just a little bit about this bull for those of you, and this is where the joke comes in, right, Cedric? This guy Pony Farm's Arlinda Chief was born in 1962. He had 16,000, more than 16,000 daughters, 500,000 granddaughters, and more than 2 million daughters. In 1990, this single bull accounted for more than 12% of the genes in the North American Holstein population. So when Cedric started to talk to me about AI, I always thought he was talking about artificial insemination, but it's taken me a few years now to really appreciate that AI, of course, is something, could be something else, and that's artificial intelligence. But for me, AI will always be artificial insemination. And all of this was possible because of the advances, scientific advances in freezing of sperm and in doing AI. And you can imagine if you are spreading these genes, if 12% of the genes alleles in the current Holstein populations can trace back to one bull, you can imagine if that bull carried a genetic defect, what impact that might have on the current population. So just quickly, we were able to do beautiful things that had not been done. We were able to do the first really phasing, detail phasing by genotyping with a 50,000 SNP chip. We could go back and reconstruct the haplotypes. Remember, Mark was a son of Chief. So we had two other, also many sons of Mark. So these is 92 progeny tested sons of Mark. And we could phase the haplotypes in all of these individuals because we had SNP genotyping and resequencing of both Mark and his father, Walkway Chief, Arlinda, Pony Farm's Arlinda Chief. Okay, so this is a look again at cattle chromosome 29, and you can see the haplotypes very clearly. One haplotype is all red across the top. The other major haplotype is the blue haplotype, and all these other individual bulls represent recombinants on cattle chromosome 29. And well, how did I go that way? I meant to go this way. And again, with genotyping, just a visual inspection, if you look at the descendants, chief descendants, this is looking 6 generations ahead of Pawnee Farm's Arlinda Chief. In the Australian population, Australian Holstein bull population, you can see on chromosome 29, most of these progeny tested sons have segments from one of the two haplotypes, the red haplotype and not the blue haplotype. And this is how you can almost visualize how this selection has occurred. Since these were progeny tested sons, this is not a random distribution. These are already the ones that are selected as replacement bulls for the next generation of breeding. And make a long story short, you can go through, you can find 49 areas that are, 49 areas of the genome where there is evidence for selection, and we can identify candidate genes. And that was all very nice, but that leads to the interesting part of the story in that in 2011, Paul Van Reyden at the USDA Beltsville identified a haplotype using SNP genotypes that was responsible for 1,000 spontaneous abortions in the United States and more than 500,000 abortions worldwide over a 35-year period. That haplotype was mapped to a 7 megabase region on chromosome 5, and it was traced back to who? It was traced back to Pawnee Farm's Arlinda Chief, that bull that we had resequenced. Now, in follow-up studies, Paul and his colleagues genotyped 70,000 individuals. This is just for a haplotype. They didn't have any idea these were just sort of random SNPs that were used on the SNP 50,000 SNP genotyping panel at the time. And they were able to fine map this to a 3 megabase region. So they reduced the region by half. That region contained 39 SNPs. And Paul called me up one afternoon because he knew that we had sequenced Chief and Mark because USDA had funded this project. And he asked me, he says, well, all of this maps back to a single bull, and you've sequenced the bull. Do you think that you could identify the mutation in that bull? I said, well, sure, why not? It only took a day to do this. We went back to the data, and we used Anavar, some very, standard tools at the time, and we identified, we classified the mutations within this 3 million base critical region. There were more than 1,500 SNPs, but to just make the long story short here, there were only 3 of those that were in exonic sequences involving one annotated gene called APAF1 and another unannotated gene with an unknown function. And so if we look more closely at all of those mutations, we see that there was one single mutation in APAF1, which was a stop gain mutation, a nonsense mutation in exon 11, a T to C change that caused the stop codon in exon 11 of the APAF1 gene. And if you looked at that mutation had 100% concordance going back to the previously identified animal. So even though there were this group of SNPs that you see here, only one of them had perfect concordance with the phenotype. And I say, oh, okay, that's very nice. What is this gene? And this gene, APAF1, turned out to be quite an important gene. I'll talk to you about the function, but the stop codon, as I mentioned, was in exon 11. That stop codon truncated the protein. About 50% of that protein was not translated, so it took about half of it took about 670 amino acids out of the 1,248 amino acid full-length APA F1 protein. And so this was a really, really good, strong candidate gene. And then we used the tools of comparative genomics, okay? Then we went to I said, okay, what does this APA-F1 gene do? And we were able to go to the mouse knockout database, and we were able to find that this particular gene was involved in the apoptosis pathway and was a critical gene in early development for the proper developmental pathways leading to notochord and central nervous system development. So mice that had a knockout in the APA-F1 gene, if you look in the middle chart, had completely abnormal development of their central nervous system, and all of these mice basically died in the first trimester. So the pattern fit very well. You had low reproductive efficiency in the animals with the HH1 haplotype, okay? And you found in the population, let me go through now the genotyping data. We went back and did confirmatory genotyping on about 250,000 Holsteins. 5,299 were heterozygotes, but there were 0 homozygotes for this glutamine stop codon at 579 that knocked out the glutamine, and then the rest of the chain, no homozygotes. You would have expected about 100 and no homozygotes at the population level. And so here, to just finish up the story, this mutation was responsible at least 10 years ago. I don't know what the numbers are now, since things have been cleaned up since then, but for 525,000 spontaneous abortions over 35 years and over $420 million in losses. This is probably a single gene with perhaps arguably the greatest economic impact in the dairy cattle industry that has ever been discovered. And the likely causal mutation for this, we believe the evidence is really strong, has now been eliminated from breeding these breeding bulls by genetic testing. So this is a long walk, okay, from where we were 40 years ago to a very nice example of how genetics and genomics has led us to the discovery of an important trait, which has had elimination of this mutation has had very important economic consequences for the dairy industry worldwide. And I just want to acknowledge here in the last few minutes the collaborators for the latter part of this study, particularly my collaborators in Australia, Mike Goddard, Ben Hayes, who many of you know, Hans Detweiler, and Iona McLeod, and Dennis Larkin, and Heather Adams and Miri Cohen-Zinder, who led this work in my lab, and the folks at USDA BARC, who discovered the HH1 haplotype, Tad Sondergaard, Kurt Van Tassel, Paul Radin, and George Wiggins. So that's a long story. Thank you very much. And I'm here to take your questions.
Haris, thank you so much. This was an awesome talk. This was really great. And this last part, bringing together genetics, comparative genomics, and economy is really bringing everything together. You have convinced us of the relevance of AI, especially for our breakfast. And may I suggest that Gengis Khan would have been a better name for Mark, no? Given so also I have to say I had this brainwave when you were showing this Earth BioGenome. You really are laying out the foundation for the United Nations of genomics. Really, that's what is going to be left of this in the decades, if centuries to come. It's quite amazing. So thank you very much for sharing all of these things with us. And I'll be really happy to take any questions. So if you want to ask a question, if it's not too messy, just unmute yourself and ask your question. Many of you have asked permission to Jose to record. You don't need to. We will be posting everything on YouTube within a few hours. So if you want to unmute yourself, anyone? In the meantime, maybe I can start with the first question for Haris. So you mentioned Nagoya Agreement, which is of course very, very relevant for these multiple sequencing efforts spread across the world. But as you know, something is coming out with artificial intelligence these days, which is that the knowledge extracted from books, songs, musics, and all these kind of things amounts to some kind of copyright infringing. And there are a lot of discussion about this. How do you see the future of Nagoya Agreement with respect to all the genomes that are being collected all around the world? If all of these things get combined using artificial intelligence to generate new synthetic biology, does this mean that Nagoya will need to be renegotiated, taking into account the leverage of AI?
Yeah, great question. This is very, very nuanced and complicated issue for the Earth BioGenome Project. And, we're kind of at a crossroads in the project, and I think in genome sequencing worldwide, where there's this tension between, the funding agencies who want to protect their the intellectual property rights around their biodiversity. And of course, the Earth BioGenome Project is an open project, right? And so we require that all to be counted as part of EBP, as part of the EBP, you're expected to submit your sequences, your assemblies, and annotations to an INSDC database, right? And so once you do that, the sequence is in the public domain. And EBP affiliates, we're actually working on an IP policy now. As I mentioned, all the projects have signed on to respecting the Nagoya protocols. But at the same time, there are many countries around the world that are very fearful that once the sequences are in the public domain, that others can utilize those, for the creation of intellectual property, and benefit sharing will be will be an issue in those cases where maybe thousands of sequences could have gone into an analysis to produce some useful IP. And so the tension in the project is that there are some projects that are going to go completely offline and they won't be part of the EBP because they're going to protect the sequences that they generate in their country. And one example is the New Zealand project. They are completely private. They are not submitting their sequences to the INSDC databases, one of the INSDC databases, unless they want to publish, on those sequences. So this is a tension, Cedric, because once it's out in the public domain, anybody, it's free. It's free to use. And part of what Nagoya is trying to implement, which has been very difficult, is the tracking of the use of those sequences. And that there's no answer, there's no solution to that. People have been talking about blockchain and so on, but none of the databases are going to do that. And so, the Convention on Biological Diversity, the new Kunming protocols that are coming out are dealing with digital sequence information and the use of digital sequence information. And to answer your question, I think it will have to take into account AI and the possible downstream use by people using AI to extract information that might lead to intellectual property. And so there's a lot of uncertainty. There are no answers to this. It's a very active area in international policy. And if you're interested in this, you have to follow what's going on in CBD. The digital sequence, the network, there's one that's headed by Amber Schultz, who's in Germany, and EBP is part of that, and we have representation in that group. We have representatives attend the CBD, all of the meetings to represent our project, which is, encouraging the use of encouraging open data and having this data being freely available to all. But with that comes concerns that very few people who have the tools will be able to use that information for in a new form of biocolonialism, if you will, if I can use that term. And it's not a trivial issue, and it's one that I think CBD and Nagoya will have to deal with. AI will be critical.
Thank you, Harris, for this very thoughtful question. We are very interested in this because a big chunk of NF Core now is dedicated to the study of provenance. Provenance is essential. Whenever you do a diagnostic, you need to be able to say that this piece of prediction is based on the combination of this software, this thing, and this thing. And I'm deeply convinced that we now need to create a new provenance for artificial intelligence to make sure that any conclusion reached by an AI system is factually based, can be factually assigned to specific inputs that were used for the training. It's very important, not only for commercial reasons, but also for obvious conceptual reasons. We need to understand how knowledge is being built. And I find it interesting that these two things will probably align towards more fairness.
Well, that's beautiful point because provenance in the EBP is incredibly important. So
Yeah.
every sample, we are using the Darwin Core Standards for our project. Every sample has provenance. We know we have recommendations and guidelines for the collection of metadata with the sample and attribution of who collected the sample, when it was collected, where it was collected. And all of that eventually can go into a system, which is the parallel to the system that you're talking about, which is traceability, trackability, and then accountability on the back end. So it's very interesting that your little pieces of code are almost like,
Yeah. They're alive.
A species. Yeah.
Yes.
It's really interesting.
Do we have any question for Harris? Please unmute yourself if you want to ask any question. I could keep asking questions to Harris for the rest of the hour, but all of our guests are also welcome to jump
You
in.
must have some old friends out there somewhere in the crowd. The 38 people.
Just a quick question
Hey,
about the current. Hi, I'm Olga. I'm also in the California, US right now.
yeah, where are you?
And I'm in Alameda, the East Bay of the San Francisco
Okay,
Bay Area.
only an
Yeah.
hour from me.
Okay, great. Yeah. But yeah, I founded a nonprofit research institute for marine genomics where we annotate proteins of unknown function. So I'm very interested in the EBP. And I was wondering for your slide on um, the showing that 80% of the genomes that have been sequenced but don't live up to the standards by the EBP, are those genomes in NCBI ensemble? Like, where are they coming from?
Yes, they're in one of the INSDC bases. They're basically in all in NCBI. We take them from ABI and NC. Did I say ABI? I'm still thinking of the old sequencing. NCBI and those are scooped for right into GOAT, the GOAT
Okay. And
system.
so if someone wants to sequence a new genome, should they even
Yeah.
bother or should they just join the EBP?
Well, what you should do is use the duplication tracking tool in the GOAT database. So if you want to know,
Mm,
there are lots of ways to know, but if you go to GOAT, I think it's the most direct way to find out if the genome you're interested in has been sequenced and whether it's sequenced to EBP standard. And you can find that out in a very easy, straightforward manner, a very easy search. Now, if you have a long list, like there are new projects that are coming up and people make a list of 300 targeted species, and then you want to know, how do I do that? You can also do that, but you have to submit the list to us, that
mm,
your target list, and then we can produce a duplication report. We can tell you, has another sample been collected? Is it in progress? Where is it in progress? Is it in sequencing progress? Has it been sequenced, been completed, and it's an annotation? All of those can be taken out of the GOAT, you can get that information out of the GOAT system. Now, one of the things that has really confused people, and I think it's really great that you asked this question, Olga, because one of the great confusions is people will go and they will see, oh, my species has already been done because it's on the list of VGP. That's not necessarily the case.
mm,
There are differences. We're trying to get across the point of a target list. So, like the Darwin Tree of Life Project has submitted like 60,000 species, a list of
mm,
all the species in the UK and Ireland. That's their target. That doesn't mean that a sample has been collected from
mm,
60,000 species. So you can, if you find your list on somebody else's target list, doesn't mean that it's been collected, doesn't mean that sequencing is in progress, and so on. But you can get that information. You can
mhm.
see where, if a sample has been collected and whether sequencing is in progress from the GOAT database. Okay, so you can see if it is on somebody's target list, but you can also see where it is in the sequencing pipeline. And so don't be confused if you find, you go there and you say, oh, that's on the Darwin Tree of Life list. They only have about 6,000 species that they have collected now for sequencing. So that's only 10% of all the species in UK and Ireland. There's lots more there.
Oh, all right, great, thanks. Yeah, we're working on Arctic clams. That's the one species.
Beautiful,
Great. Thank you
beautiful.
so much for your talk.
You work with Bodega Bay at all out there? The people out in the Bodega Bay
Not
Marine
yet.
Lab?
No,
Okay.
one of our board members is from the Stanford Hopkins Marine Station.
Okay.
But the data comes from a collaborator who really independently got PacBio and Illumina to sponsor a bunch of sequencing and convince the Canadian government to let him go uh, collect these samples. But the goal is to cure his father's neuropathy because Arctic clams are able to tolerate a toxic compound that in humans causes neuropathy.
Oh.
Fantastic.
Yeah.
I'm going to eat more Arctic clams, that's for sure.
Yeah, they're also yeah, there's a crop or what's the term? But yeah, also a food.
Yeah.
Will not be difficult, no? I agree.
Thank you for your question. Yeah.
Thank you.
Do we have any other question? Thanks a lot for this great question, Olga. Do you have any other question for Harris?
I do have a
If you
question.
want to ask, go ahead.
Thank you.
Unmute yourself.
Yeah. Harris, nice talk. So
Thank you.
you said your
Oh, it's Robin.
so you said your project was going to be 1.8 million sequences in 10 years. So how likely is that going to happen in 10 years? And what's your next project going to be?
My next project will be my funeral, I think. But but how likely is it in 10 years? I would say right now I'd give it less than 50-50. But you know, when we designed the project, we designed it as a moonshot. So when John Kennedy said we're going to send a man to the moon in 10 years, they didn't have the technology to do it, right? They didn't have the methods, they didn't have the engineering, they didn't have what they needed, but he set that as a goal. So we set the goal for 10 years and we tried to map out a strategy that would work in 10 years. Now, I think we have a strategy that will work, but I think it's more about scaling, about getting more labs around the world, about decentralizing sequencing. We're going to need about 25 centers around the world to be doing very high throughput genomes, producing literally hundreds of genomes a week to reach that goal of 1.8 million. Once we get to that 25, that scale, those 25, like those 25, you know, the regional nodes and those individual projects in the node, once we get there, it's going to be scaling very rapidly. Will we reach 1.8 million in 10 years? I don't know. We can certainly reach 150,000 in the next 4 years by 2030. But after that, we've got 1.64 million to go. We need better technology. We need better computational tools. We need everything that's necessary in the pipeline to scale to 1.8 million to hit the goal in 10 years, including the funding, which is not trivial because we're talking about several $billion to get there. So Robin, you know me. Robin was a postdoc in my lab for a long time. How long, Robin? Too long? 6 years?
Just long enough, 6 years. Yeah.
6 years, and his wife was a graduate student, and his picture, I think, was on one of those slides. Robin knows that I am an incurable optimist, and I don't easily take no for an answer. Is it overly ambitious? Is it optimistic? Yes, but we've got a lot of people that have bought into it and are thinking hard on how to do it, and are willing to go and work hard to raise the funding and do the organizational work necessary to do it. So Robin, I hope that is an honest answer to your question. I don't know if we'll get there in 10 years, but I can assure you one thing, we're going to get there. I don't know when exactly, but we'll reach the target eventually.
Thank you.
Can I bounce on this, Harris? Because you're right in the eye of the cyclone, and it looks like Moore's law is accelerating in AI, and we see the dropping cost of the training of LLM and this kind of thing. That was not expected, and that's a change of technology. When it comes to sequencing, it's really, really difficult to figure out Moore's law because there are switches from short reads to long reads, and the halving time is very, very bumpy. But what is your gut feeling? Could it accelerate? Could the doubling time, the improvement of the technology become even faster than Moore's law? Or have you factored this in? Do you think it will have an impact?
Actually, we did factor it in. We made several assumptions in that paper about reduction in cost and throughput to reach the goal in 10 years. And, with the Revio, the technology is there today. If you had enough machines now, you could do this, and it's not a lot of machines, that you would need. Certainly 100 Revios would probably do the job. If you do, each Revio can do about 2,000 genomes a year. So, 100 Revios or less around the world would certainly do it. The major limitation, Cedric, is really not. I don't think it's the technology. I don't really think it's computational to do the assemblies. I think at least to the assembly part, annotation, yes, that's a problem. But the assembly part, no. I think the assembly algorithms are good enough and efficient now to do it. The big limitation is on the front end, the collection of the samples. It costs more now in some cases to collect the samples than it does to sequence and assemble, right? Which is really amazing if you think about it. But a millionfold cost reduction in sequencing over the last, 20 years or so has gotten us to where we are. And so what we have been spending a lot of our time on, Cedric, is trying to scale the collection, taxonomic identification, vouchering, biobanking of all of these samples. And this involves, museums, big collections, global networks like the Global Genome Biodiversity Network, and figuring out where things are, how you can get them, how you can collect them so that high molecular weight DNA can be extracted so that you can do the long read sequencing. And what about all of these single cell eukaryotes, right? Which is thousands. We don't even know really how many there are, just the named ones, and it's still really a relatively small number to what actually exists there. But we need technological improvements in the sequencing of single cell eukaryote, non-culturable single cell eukaryotes. Otherwise, it's going to be very difficult to reach our target. So the front end is critical. Small samples and single cells are critical. And I think on the back end, annotation, AI, especially for annotation, it's still a bottleneck right now at EBI, at NCBI. They're trying to decentralize and have their software being used by local laboratories, but it's still quite we're still quite behind. So like in the VGP, for example, only half the genomes that have been produced have been annotated, even though the data sets are already there to do the annotation. And then, of course, making sense of the data, Cedric, and doing all the kinds of things that the folks on this call want to do, which is to deepen their biological understanding and create useful new technologies that can be used for animal improvement of agriculture, for example. So yeah, I think it's possible. I don't think it's a technological. I think it will increase. Will we get cheaper? Yeah. How is it going to get cheaper? We're going to have flow cells that have more channels. You go from 2 million to 4 million to 8 million to 16 million to 32 million. I mean, I don't know how many channels they're going to end up being wave guided. Channels they can be able to get on a single chip. But that certainly has made a big difference already, and I think that's going to continue. That technology will continue to improve and give us greater efficiencies on the sequencing, right? It'll get faster, it'll get cheaper, but it's no longer the limiting factor. The limiting factor is collection, DNA extraction from very difficult samples and plants and things that are really hard to get DNA from. And so the biggest challenge is right there.
Yeah,
And funding.
we had in Roderick organized here in Catalonia, Roderick Grigo organized here in Catalonia, or was part of a major sample collecting effort in the lakes, in the lakes of the Pyrenees, which I'm sure he will try to get you there next time you come around. And so thanks a lot, Harris. We are well past the hour. This has been a very, very successful turnout today. I want to thank you personally for giving us an insight into something that is worldwide collective. This is not the easiest patch of history we are going through these days. And it is so nice to see a project that is showing the whole world having to work together because, we live in the same planet. And so this showed very nicely. So thank you very much for this.
Well said. Well, it's a privilege to be part of this thing. And it's even a greater privilege to be leading so many distinguished scientists and young scientists around the world. I mean, I think, Cedric, the most exciting thing is I'll shut up and get off and let you get on to Chris Tuggle. But I think the most exciting thing is the young people that this has brought into the field. People who don't recognize the constraints, like, you know, Robin, he's still old enough to remember, oh, sequencing one genome was a big deal. And so we bring to the table, you know, these past biases and doubts. But young people come in and just say, why not? Yeah, you know, a thesis maybe was a gene when I was a student, or my first graduate students, then it became a few genes, and then it became, you know, eventually a genome. And now, you know, Mark Blackser just graduated, a graduate student who did like, you know, 150 Lepidopteran genomes. I mean, it's just mind-blowing. And young people just don't bring the baggage. They just say, sure, why not? Yeah, 1 million, it's great. It's going to save the world. We're going to be able to do conservation. We're going to be able to improve the bioeconomy. I mean, they're really excited. And the thing that's most gratifying to me is the number of young people who have been drawn to this project, who will be the next generation of leaders, because this, as Robin has pointed on, will likely go on for many years beyond my career and my tenure as the chair of this project.
Your telomeres are still long, so we have time to go. Thank you so much, Harris. This was so interesting and inspirational, and really thanks a lot. I want to point out that a lot of the Darwin Tree of Life analyses are carried out using NF-core tools or NF-core compliant tools. Darwin Tree of Life maintain their own collection of tools that are developed by the NF-core standards, and they have actually a tool page that is somehow loosely affiliated with the NF-core standard page. Maybe somebody is going to paste this on the Slack. And so this is really a project we consider a brother project. And so we are very, very excited and interesting with all the stuff they do off there. So I don't want to take much more of your time. We are way over the hour. So thank you very much.
Thanks. I haven't talked about animal genetics in more than 10 years, so I appreciate the opportunity to do that. I always talk about chromosome evolution, but I thought this group might like a retrospective. So
I think everybody was interested, and that was really a nice way of putting all of these things together. Jose, I think you have an important announcement to make for NF-core, and we are going to wrap up on this with Francesca just after.
Yes, sure. So thanks, David. Yeah, I just want to announce that the spring NF-core hackathon is taking place from the 24th of March till the 26th of March. And usually there are 2 NF-core hackathons during the year, one that is completely on site during the Nextflow Summit on October in Barcelona, and one that it's hybrid. And during the spring, and this is the one that it's taking place in March and that you can attend either online or in a local site. For instance, we are hosting one of these local sites at the CRG in Barcelona, but there are plenty of sites all around the world, so you can check the list in the NF-core site. Actually, I'm going to share a message now so that you can check it out. And yes, we think it's a very nice opportunity for anyone interested in getting in touch and contributing with the NF-core community. So we are looking forward to seeing you during the hackathon. And now I will pass the stage to Francesca, who is part of the coordination team of the animal-genomics, NF-core special interest group, and she will announce the March meeting. So all yours, Francesca.
Thank you so much, Jose. And thank you again, Harris, for the beautiful and engaging talk. Before we close, as Harris already mentioned, we will have Dr. Christopher Taggle in our next session on March 19th at 4 p.m. Central European Time. And he will present an overview of the work that he is doing on single-cell research. And since it promises to be a very interesting session, we look forward to see you all next time. Thank you so much again for joining, and see you next month.
Thank you all. Thanks, Harris. Bye-bye.
Bye.
Ciao.
Thank you all.